Hi there, welcome to our IB Biology revision series on section A2.3 viruses. This is a higher level topic. So for section A2.3.3 and section A2.3.4, you need to be aware about the lytic cycle of a virus and also the lysogenic cycle of a virus. So before we look at the lysogenic cycle and lytic cycle, let's look at the life cycle of a virus. So to reproduce, all viruses must attach to a site on a specific host cell, incorporate their genetic material into the cytoplasm of the host cell, and then utilize the host cell processes to produce components of themselves. They assemble the viral components into new functioning virus entities and then release the new entities into the host cell environment. So the lytic cycle. This is referred to as lytic because new virus particles are released by the lysis or rupturing of the cell membrane by an enzyme called lysozyme. This enzyme is coded for the bacteriophage DNA, and lysis can only occur after the production of fully functional virus particles known as virons. So the diagram presented in the slide shows the stages of the lytic cycle. In phase 1, you've got attachment. Phase 2, you've got penetration. Phase 3, you've got biosynthesis, where the phage DNA directs synthesis of viral components by the host cell. Then stage 4, you've got maturation, and then stage 5, you've got the release of the virons. So, the lysogenic cycle. The lysogenic cycle does not result in the immediate release of new viruses. The DNA of the bacteriophage combines with the bacterial DNA to form a prophage. Now, in the lysogenic cycle, the viral DNA is integrated into the bacterial DNA, and as the bacterium replicates its DNA, also replicates its viral DNA as well. So basically, what you've got going on here is you've got as the bacterial DNA replicates, so does the viral DNA. So then what's happening here is the integration of the latent prophage with the bacterial DNA can result in the continual production of bacterial cells containing the prophage. The prophage is active within these cells. Once the prophage is released, then the lytic cycle is initiated. So the diagram in this slide shows the lysogenic cycle. So in stage 1, the phage attaches to the host cell and injects DNA. The phage DNA circularizes and enters the lysogenic cycle. The phage DNA in stage 3 then integrates within the bacterial chromosome by recombination, and this is where the prophage is formed. The lysogenic bacterium reproduces normally, and then what can then happen is the prophage may excise from the bacterial chromosome by another recombinant event, and then this can initiate the lytic cycle. Thank you very much for listening to this presentation on section A2.3 on our IB Biology revision series.